Today we will learn Mendel and his works. So we all know Gregor Johann Mendel. So Mendel and his works. Who was Mendel? Gregor Johann Mendel was an Austrian biologist, meteorologist, mathematician, Augustinian monk. He was the first person to lay the mathematical foundation of the science of genetics in what came to be the Mendelism. Mendel, known as the father of modern genetics, chose to study variation in plants in his monastery's 2-hectare experimental garden. So, in this slide, we can learn about Mendel's and his works. So, in this next section, we will see an overview of Mendel and his works. So, the full name is Gregor John Mendel. His father name Anton Mendel. Date of birth July 22, 1822. Place of birth Vienna, Austria. In 1840 he was graduated. In 1843 joined the Augustinian monastery at Brunn in Moravia. In 1846 attended some course on agriculture in Philosophical Academy at Brunn. In 1849 appointed as a substitute teacher in the Imperial Royal Gymnasium in Nîmes. 1851 to 53 attended some courses on mathematics and natural sciences, University of Vienna. In 1853 took the membership of Zoological Botanical Society of Vienna. 1854 to 68 appointed as a teacher of physics and natural science in higher secondary school at Brunn and continued up to 1868. In 1856 began experiment with garden pea. In 1862 became a founding member of the Brunn Natural Science Society. 1865 delivered his first lecture on garden pea experiments to Brunn Natural Science Society. 1866 experiment on plant hybridization was published in volume 4 of the Proceedings of the Natural Science Society. In 1884, 6 January, the pioneer of the classical genetics was died in Brunn. So you can see a lot of information in this slide. So we don't need to memorize all this slide, but we can give more attention in some line that he was born in Austria, which is important. Then the another important is his date of birth, which is July 22, 1822, which is similar 22. So and the main year which needs to remember is 1856 began experiment with garden pea, and we should know when the father of this genetics died, which is 1884, 6 January. The pioneer of the classical genetics was died where? Brunn. So you can see that Mendel's book here. So Mendel's methods of symbolization. English alphabets were used to Mendel to represent factor or gene or allele. First of all, he give the name factor, then it becomes gene, and then now we can say allele. To explain the dominant and recessive relationship between the alleles and to symbolize the allele constitution genotype of an organism, the post-Mendelian genetics have evolved following 3 methods of symbolization: classical, modified classical, wild type symbolization. Classical method. Dominant allele is representing by its first capital letter of a dominant trait, while the recessive allele by small letter. Genotype of homozygous tall pea plant will be capital T, and the homozygous dwarf plant is small t, small t. The genotype of heterozygous will be capital T, small t. Modified classical method. Dominant allele is representing by its first capital letter of a recessive trait, while the recessive allele by a small letter. Homozygous tall pea plant will be capital D because D for dwarf, and we will emphasize the first capital letter of recessive trait. So dwarf is recessive trait. So first word D, which if it is capital, then it will be homozygous tall triplet and homozygous dwarf plant as small d, and the genotype of heterozygous will be capital D small d. Then we will learn the wild type symbolization. Wild type by the sign plus and the mutant type by small letter of recessive allele. When out of two phenotypes, one phenotype is much common occurrence in the population, that is alternative phenotype known as wild phenotype. The phenotype which is rarely observed in the population is called the mutant phenotype. For example, in P, dwarf plant type is governed by a mutant gene small d and a tall plant type by wild type gene plus. Then we will learn some on experiment on plant hybridization. Mendel works. Before Mendel's work, several investigations or hybridists had made hybridization experiment with plants. Mendel was familiar with the work of predecessors. Mendel's approach was simple, logical, scientific, mathematical, and analytical, probably because he was well aware about the failures of his predecessors' hybrids. The pre-Mendelian investigators made observation on plants and animals as a whole, and they studied all the variation in the hybrid at a time. Mendel concentrated his attention on a particular character and at a time he studied only one character of the hybrid. Unlike other hybridists, he designed his hybridization experiments to record the number of different types of the progeny, to arrange these types of with certainty according to their separate generations, and especially to ascertain their statistical relation. Then we will learn his direct words, that experimental plan must necessarily possesses constant differentiating characters, and the hybrid of such plants must, during the flowering period, be protected from the influence of all foreign pollen grains or be easily capable of such protection. Then we will learn he choose pea plants because of easy to culture in open ground or pots, short growth period and life cycle, self-pollinating flowers of peculiar structure, contrasting heritable characters, ability to produce fertile hybrids on artificial cross pollination. So Mendel used seven characters in his experiment, which is length of stem, forms of the ripe seeds, color of the seed cotyledon, form of the ripe pods, color of the seed coat, color of the unripe pods, position of the flower. So you need to remember these seven characters because it's important and it's often come to the examination. So length of stem and three color. One color seed cotyledon, another color is seed coat, and another color is the unripe pods. And two forms of the ripe seeds and form of the ripe pods. And first one is length of stem and last one is position of flower. That's the seven characters Mendel used in his experiment. So let's see Mendel's procedure. Mendel first had to see that the parental lines of chosen plants are pure, that and he gave seed of only one type when allowed to self-fertilize. He got the pure line for each character of the seven pairs of characters of P by self-fertilizing the plant for several generations until the that started to produce all the offsprings exactly of that own kind. For example, to get a pure line for tallness of the stem, he self-fertilized a tall pea plant for several generations till the resulted offspring always produce only tall plants. Likewise, he got genetically pure variety for short stem dwarf pea plants by adopting the same technique. The two pure lines tall cross dwarf was crossed by removing the stamens for all the flowers before the anthers were mature and dusting the stigmas of both parents with pollen derived from each other. When both strains were used as male and female parents, a reciprocal cross was made. Later in the same year, the mature pods could be opened and the shape, color, etc. of the first generation pods seeds and cotyledon were recorded. In the next year, Mendel sowed the seeds of first generation to record the characteristics of stem, leaves, flowers of mature plants resulted by these seeds. These mature plants of first generation were allowed to self-fertilize, and the characters of offspring of subsequent generations were recorded. So thank you for being with us. The rest of the thing will be covered in part 2.